Good morning. Thank you for the introduction. So this is the title. Probably should read Mechanism of Chemoresistant in Bladder Cancer and Prediction of Chemoresistant and Future Therapy, but it was a big title, so I just put that as a title. Just a bit of background. So I'm not a scientist. I don't know much about the topic prior to started reading this. So just forgive me if I over-explain things or I miss some things. There are a few scientists in the crowd. I'm sure they correct me as we go. So bladder cancer is common. It's the second most urological cancer. It's a very costly malignancy. And if you look at the cost analysis data, it's probably the most expensive cancer. And most of the money goes towards surveillance and diagnosis. Get to the topic of chemotherapy. It's relevant in all aspects of bladder cancer treatment, whether it's a non-muscle invasive bladder cancer or more advanced disease. I won't talk much about intravesical chemotherapy as we got a review coming up. Hopefully, we'll see it in the publication. The next three topics, chemotherapy is quite relevant. In North America, we don't see much neoadjuvant chemotherapy, but there is a level 1 evidence, as you all aware. It improves survival, 5 to 8% absolute survival for people who receive neoadjuvant chemotherapy. Adjuvant chemotherapy, the data is more slim. There is some suggestion is helpful, but again, we won't discuss that. And then chemotherapy for systemic disease, which is the mainstay of treatment in patients who present with metastatic disease. So what I like to talk about is why, despite these chemotherapy treatments, we still having high incidence of recurrence, progression, and death from advanced bladder cancer. So as you can see, the survival is abysmal. So when patients present with metastatic disease, the median survival is very short. The chemotherapy agents, based on randomized trial, which is more than one around, the multi-regimen treatment is better than single drug treatment. This study just outlines that one of the first studies to look at it, and you can see combination treatment is certainly better than cisplatin alone. What I like to point out is the last line, and you can see the toxicity is quite high. So in this series, 5 patients died from MVAC treatment alone, and 2 of the people who crossed over from cisplatin alone to MVAC again died related to the toxicity of the treatment. So in an attempt to improve that, the gemcitabine cisplatin regime came along. And this paper, along with a couple of other smaller randomized papers, shows that it's equivalent as far as the cancer control is concerned. But gemcis is much better tolerated. So we have a chemotherapy regimen. It's pretty much extremely toxic. Gemcitabine cisplatin regime has improved the situation, but it still remains a toxic treatment. We're offering it to the patient who have very limited lifespan, and we're making things worse by making, increasing toxicity. So ideally, we want to offer it to people who would benefit from it, and we try to at least we can justify the toxicity, and if further down the line we can come up with less toxic treatment, that'll be ideal. The possible reason that treatment doesn't work as well as it should is this inherent ability of the urothelial cancer to be resistant to chemotherapy. Some resistance develop after exposure to chemotherapy, and some cells are just de novo resistant, so they are born that way. And so because cisplatin is the main drug between the two known regimen, I'd like to use that as an example, provide some explanation for mechanism of resistance, and then focus on future and also for diagnosis of chemoresistance as a whole. So this is the organized part of the talk, and then things get a bit less organized. So cisplatin works basically by reacting with both DNA, RNA, and also with protein. But it's the formation of the adducts is the cause of apoptosis. When it enters the cells, it loses those chloride molecules, becomes hydrolyzed. Then around here, it forms dimer with the nitrogen within the purine molecules within the DNA. They can form adduct between the strands of DNA or across the strands. And if the cell cannot repair this, then the cell will go toward apoptosis. And by generating this cell death, that's how cancer cells go and die. So in order for it to be able to do its job, it needs to enter the cell. Typically, it uses this transporter, which is a copper-dependent transporter, to enter the cell. As long as it stays within the cell and can get to the DNA, then the dimers are formed and the business as usual. Cancer cells seem to have ability, which we've known about a long time, and it's been seen in a variety of cancer cells, and it's not special to urothelial cancer, to efflux the cisplatin out of the cells. So 2 transporters have been identified that achieve that. One is these guys here. They are P-glycoprotein, which is, if you look at the literature, they go back to 80s and 90s. We've known about them for a long time. They seem to be upregulated in many cancer cell lines that are resistant to cisplatin. Their role in urothelial cancer is not well known, but it's speculated they play an important role as well. The other molecule it's a long name for it, but so CMOAT for short. It's another glycoprotein, which is, we have more information about it, this guy here. So again, it's an ATP-dependent efflux transporter pump that takes the cisplatin that's entered the cells out of the cells. It uses usually cisplatin conjugated with glutathione to efflux it out of the cells. In cell models, we've noticed that when the concentration or expression of this molecule is high, the cell develops chemoresistance. If we come and try to target that molecule, again, this is all cells, lab studies. If we come and disable this molecule in the chemoresistant cells, the cells seem to be sensitive to cisplatin again. So it's a possible future target. I briefly mentioned glutathione. It basically belongs to a group of intracellular thiols. All of these have been associated with increase in the situation of chemoresistant. It's possible that cancer cells use these molecules to protect themselves. Metallothionein is one of them. It's basically a molecule that detoxifies the heavy metal ion. Cisplatin being platinum gets deactivated. Our normal cells express this and have a high concentration in liver and kidney and intestine, but urothelial cells seems to have adapted as well, and that's another way they fight the cisplatin. I mentioned glutathione. Again, earlier studies put a lot of emphasis on that, and it seems to be increasing expression itself and also all the enzymes associated with this metabolism. But we haven't really figured out how and why we can counteract that problem. Again, cell studies shown if we control glutathione concentration within the microenvironment of the tumor, we can improve the chemosensitivity. So that's as far as the pharmacodynamic goes, as far as the cisplatin gets into the cell. Once it gets into the cell, as I mentioned before, it forms a dimer across with the DNA. Then based on these guys, the nuclear excision repair enzymes, the cell repairs itself and survives, or can repair itself and goes through apoptosis, which we'll cover shortly. So from those enzymes, this guy here, so excision repair complementing complex with XPF is quite important. So again, I did medical school 10 years ago, and I haven't come across this since then, but a lot of literature supports that it plays a role. So this is a study. It looked at patients with advanced cancer, locally advanced or metastatic disease who had chemotherapy agents. And they went and looked at the expression of this protein, which is that excision repair enzyme. And it was noticed when the expression is increased, the cells survive longer, the patient does worse, and the disease comes back faster. So it's speculated that these cancer cells have adapted to repair the DNA damage caused by cisplatin by using the excision repair enzyme and survive. So this might be a future target too. Other excision repair molecules have been implicated. Sorry about too much names and abbreviation. That's the nature of the talk. XPC is one of them. It seems to be downregulated in chemoresistant cells, and this is related to epigenetic changes with acetylation of the gene. And again, if we can turn that off somehow and return the XPC concentration to normal, that might be a method of counteracting it. Mismatch repair proteins have been indicated. When we say mismatch repair, you essentially remember colon cancer. So a lot of work has been done with cisplatin in colon cancer situation with that enzyme. Very limited data from urothelial cancer, but that's a possible candidate. So what happens next? Cell is damaged. We're going down the pathway of apoptosis. Just a brief reminder, so we had intrinsic pathway and extrinsic pathway. With the intrinsic pathway, the Bcl-2 families have been well studied. Two group of proteins, two broad group fall within this category. So we have the pro-apoptotic, meaning pushing the cells toward the cell death, and anti-apoptotic, which counteract that. It's the balance between these two proteins that will affect where we're going to go. So once the balance goes toward the cell death, so this guy here, it's an important player. It causes release of cytochrome C from the mitochondria. The capsases get activated, and the cell dies. Obviously, the balance is quite fine, and you can see multiple checkpoints along the line that can affect the outcome. But this is a very simplistic overview. So just like to briefly talk about Bcl-2. It's been associated with worse outcome when it's overexpressed in cancer cell line, whether in the lab or clinical data. It's been shown if we switch it off, either using vector viruses to turn the expression off, or use a small interfering RNA, we can improve the chemosensitivity. So that's again possible future target. I'd like to talk briefly about a few other proteins which I don't know much about them, but I'll try to explain. So this guy down there, SMAC Diablo, it's a small mitochondria protein, and its level is downregulated in chemoresistant cells. Very little data in the literature about it. I have to look at the very hardcore science papers, but just briefly wanted to mention that. It functions by deactivating this protein here, which is a major anti-apoptotic protein. So it takes that out of the circle and pushes the cell toward the cell death. That might be another target, and it's been associated with chemoresistance. P53, don't want to talk about that much. It's been associated with a lot of cancers. There are a lot of papers indicating in bladder cancer particularly is associated with poor outcome. The data on prediction of chemosensitivity or chemoresistant is very mixed, and multiple papers have gone for and against it. So the question regarding P53 remains unknown. It's quite further down, and it's possible that other pathways before that are the dictating mechanism, and that's why the P53 results are so variable. Two other proteins, we're going to finish with proteins soon. Two other proteins have been associated with chemoresistant, the IP3R1 and calcium binding protein S100. Both of them involved with calcium metabolism, and their level seems to be lower, their gene expression is reduced when there's chemoresistant. So they might play a role too. So that was intrinsic apoptotic pathway, and we have the extrinsic apoptotic pathway. So I'll just put that there. They don't play much role in chemoresistance, but it's possible that if we can activate them as separate from the defected intrinsic chemoresistant apoptotic mechanism, we can overcome the chemoresistance that's observed. So that's something that to keep in mind. We cannot talk about cancer and not talk about growth factors. There is good data from their use in renal cancer and few data on bladder cancer. Briefly discuss them. So epithelial growth factor receptor, it's been associated with upregulation. There's a lot of pathway down the line. I want you to focus on these two, the AKT and ERK, basically pro-survival anti-apoptotic proteins. So there is limited data available. If we switch off them, or if we block the EGFR receptors, which is upregulated in chemoresistant cells, we might be able to improve chemosensitivity. So another target. I've lost count how many targets we discuss, and I'll get to this in my discussion. We mentioned AKT. It's a player somewhere within the line. majority of the cancers who are aggressive, who are chemoresistant, have upregulation of AKT. Just I'd like you to focus on these two arrows here. So these are our major pro-apoptotic proteins, and AKT seems to have a negative blocking effect on them. So that's why it's been implicated in chemoresistance. But most of the stuff we discussed is further up the chain, that other mechanism called increasing AKT in the first place. So last of the growth receptor and growth factors is the fibroblast growth factor. Probably that's where the next thing's going to happen in bladder cancer. It's definitely been shown to be upregulated in aggressive tumors, and it's been shown if we cause its expression by vectors or other modes in a chemosensitive cancer cell line, they become chemoresistant. Again, it shows that it works through increasing AKT and other signal mechanisms which promote survival. So switching that off or targeting that might have a role. There are proteins that we can use to target FGFR, and they have been associated with improving outcome of chemotherapy, but there's very little clinical data to support that. Okay, handful more proteins and we'll stop. So when you search for chemoresistance in bladder cancer, you get a lot of papers coming up, and every group has done gene expression profiling, some molecular study, and found 1 or 2 or 3 or 10 proteins that have been associated with the chemoresistance. So it's very difficult to try to sift out which one is, as we heard a few weeks ago, which one is the top guy, which one is the guy who's deciding for everyone else, the protein that's making most difference, and which one are just the soldiers. So these two proteins have been both associated with bad outcome and chemoresistant, and there could be a potential target. This guy here is another EGFR protein itself. It heterodimerizes with other EGFR receptors, and same pattern that we heard so far. It increases these ERK, AKT, all have a pro-survival mechanisms. And so that's why it's considered to be a target. The nuclear factor kappa is a transcriptive factor and is associated with many cancers and might be a potential target. Briefly, so epigenetic changes for residents and for my fellows who don't know what that is, I had to look that up, is basically changes in expression of a DNA without changing the coding. So whether the DNA is hyperacetylated or hypermethylated, and that seems to affect its transcription. We already talked about histone deacetylases with the XPC gene, and that plays a role. We also, the microRNA, which is a small RNA molecule that affects the transcription of other mRNAs. And this protein or this RNA seems to be changed in an epigenetic manner, and it's possible that changing or targeting those changes might help us with the chemosensitivity. So that's the organized part of the talk. As far as the cisplatin resistance and the mechanism goes, I'd like to mention a few other players. When I came here, Dr. Gliv took us out for lunch and talked about Clusterin, and we just looked at him blank. So me and John have never heard of it before. But if you look at the cancer literature, it's I did talk to him afterward about it once I read about it. But you see Clusterin mentioned everywhere. So it's a protein. It's been discovered in prostate cancer setting of chemoresistant cells. It's been associated with poor outcome and advanced disease and metastatic cancer in pretty much most cancers that have been studied. It causes broad-based resistant chemotherapeutic agent, and for whatever reasons, it's a protein chaperone, so that's the key word. Dr. Gliv is not here, but it's one of his favorite words. It's essentially like a heat shock protein. What it does is it stabilizes this complex. As a result, blocks back to going to mitochondria, to causing the release of cytochrome, to causing cell death. So it is quite an important protein, but I'm not quite sure with my limited understanding whether this is the protein or is it the only one. Just a brief description. So normally lives in the nucleus. When it's in the nucleus, it causes apoptosis, but for whatever reason, it ends up in the cytosol due to stress, radiation, chemotherapy, etc. The concentration goes high, and then plays this negative role to apoptosis. So the cell survives, and survival of a cancer cells cannot be a good thing. It's one of the first papers that described it. I don't get into the details. Initially, it was thought it's controlled by androgen receptors and suppressed or overexpressed, but that theory has been thrown out. The last of our proteins, this is definitely going to be the case. So empirin and survivin, again, similar manner to, they are not part of any specific pathways. They have just been from these studies, these have been noticed to be increased in setting of cancers. Just briefly talk about them. So empirin sits up here, so it's quite higher up the chain. It's a matrix metalloproteinase. It works by activating on through this guy and through all those pathways. Again, same pattern that we've seen, and causes survival. So we could potentially obstruct it at anywhere along the chain and achieve blocking its function. Survivin is the same. Again, a protein that directly blocks our capsases. So no matter what pathways are activated, if survivin is there, as its name says, it promotes survival of the cancer cell. So that might be a future target. In a very nice study that here, these guys demonstrated that if the cancer cells in an advanced setting had both survivin and empirin, this meant to read 2 positive, the outcome was way worse than if they were not present. So clearly it has a prognostic role and potentially a target role. So talked about cisplatin, I talked about a few other proteins. Gemcitabine is around the corner. I talked to Peter Black beforehand and we decided not to talk about the mechanism of gemcitabine because it becomes quite extensive. And just a brief summary. So it's out there, needs to get inside the cells, could be deactivated or could be activated. And anywhere along the line, the changes in these enzymes can lead to altered mechanism of function. So all of those potentially have a role. So I don't get into that more than this. So where do we go from here? We have some understanding of where chemoresistance falls. We have some understanding of what causes it. So I think the next question is prediction. So single gene markers, most of those I did talk about. These 2 here are associated with gemcitabine resistant. One is a transporter and one is an enzyme involved in the synthesis of RNA, have been used as a predictor mechanism. But considering there is something like 30,000 genes and about 1,000 of them are involved in chemoresistance, it's a very simplistic view to try to come up with one protein, try to switch it off, and voila. So that's not likely the scenario. So in an attempt to overcome that, people have tried to come up with genome-wide gene expression profiling. So typically they use PCR or DNA microarray, which I discussed briefly just because I didn't know much about it. I'd like to share that with you, how it works, and then look at the studies again. So the microarray tries to find out at the given time what proteins is the tumor cell expressing, and based on that information find out what genes are activated. And once we know that, see which one of those genes is associated with the poor outcome, whether that would be death, chemoresistance, etc., and then try to use them as a predictor and also hopefully for future targets. So the way it works, we have a tumor, so the idea is, so these guys are overactive, they're making a lot of protein, let's find out what mRNA is active. We take out that mRNA, then we transcribe it with the reverse transcriptor to a cDNA, which is complementary DNA. So this is the DNA that's pure, it doesn't have any intron, it's all active stuff. Then they label it with the fluorescence, which comes various colors. Then they have a pre-made chip, they have probes on it, the DNA probes, which that cDNA which we created from the tumor, which is already labeled, can stick to, and then we take photos with fluorescence and laser, etc. I'll describe that, and find out whether that gene was transcribed at the time. So they have the marker, they mix it up with our chip, let some time to settle. It's a very simplistic view, settle, and then see what happens. So what happens is we have our chip, which have the probes on it. These guys come and attach to it. Then once it sticks, it stays on. It's fluorescence. Then we take some pictures, and we get something like this. So green one means that gene is activated or expressed at the time. Red means it's not, or it's activated at different situation, and yellow is somewhere in between. Then they use a computer-based modeling system and some statistical information to find out whether this is a true result or false positive, etc. So back to our study. So these guys looked at only 27 tumors, but it was a landmark study at the time. It was quite a few years ago. They looked at the gene expression profiling of 28,000 genes. So big number. They used the cDNA, followed the techniques I described, and they came up with 14 genes. None of those genes are actually the one we talked about. But this is the problem with gene expression profiling. So then based on that, they come up with a model to see whether they can predict whether this tumor is going to respond or not. So the filled triangle and circle are the test data. So that's how they did their test. And then the empty ones, the hollow ones, is what the prediction was. So they came up with the model and then repeated in 22 different cancer. So this is the predictability. So when it's 100, that means they were, it's highly predictive. And so they got these guys right. So they predicted these guys will respond. I mean, some of them, the prediction number was very low, but they predicted the empty one will respond, and they predicted these guys won't respond, and they were right. They got these three wrong. So it's not perfect, but it's heading toward the right direction. Similar study was done in the cancers who were having gemcitabine and carboplatin. Few years later, same study. I mean, same technique. They used this microarray. That's the pretty picture. Those are the genes that are described. These are, again, they generated a list. Then they usually confirm the result. We can ask more intelligent people a bit later. I think they use the PCR to confirm the result, because you can't do PCR on all the genes because so many of them. And based on that, again, they came up with a model and they had a prediction score. So this system works, but based on that, you need to have the patient have had chemotherapy. You know the result of whether they did okay or not. Look at the pathology specimen to see whether there was response to chemotherapy. Then go back, look at the gene, and then say, okay, well, this gene was associated with response. Potentially this one wasn't, and then try to work it out from there. So in order to avoid that, in order to try to predict before the patient gets the chemotherapy, they came up with this concept, which is Coxen, which is basically extrapolation of the result from another cancer line. So I'll briefly talk about that. It's an important concept. So we have the NCL60 cancer line. These are a bunch of cancer lines that have been heavily investigated. The results are available publicly. We know what gene has been expressed. We know what the reaction was to certain chemotherapy agent. Most of the data is from the lab. So we have that information. Then we come and see our cancer cells, do gene expression profiling, find out what genes are active in our cancer. Now remember, we're not giving our patient chemotherapy yet. Based on that information and extrapolating from the results of the NCL60, we can come up with a model to predict whether the person is going to respond or not. So it avoids the treatment, then it's going to be futile. So these guys published the results. It's initially received a lot of attention, but so far no randomized trial, no prospective trial have been done because it just hasn't happened. So based on, these multiple papers they had, and again using that concept, coming up with the model, and then they do a test, and then they try to predict the outcome. So they did that, and again, going over the Coxen method, coming with the gene expression modeling. So this is different from gene expression profiling, which we talked about. And then they looked at some already known results of patients who've had chemotherapy and their tissues were available. They didn't look at the result, and so they tried to find out whether if they would have used Coxen model, they would have got the answer right. And the tests were quite promising. So we knew the results already, and we used it as a model to see whether it was predictive, and it was good. So it looks like it should be the way, but there are some few small problems. As I mentioned, it's a very complicated package. There are thousands of genes involved. You cannot potentially just put it down to 10 genes. So I think the picture is more complicated than that. The other thing is, as I mentioned, all of this is done in the lab. So we're completely ignoring the microenvironment of the tumor, which we heard about last week. We're ignoring the fact that the way it interacts with surrounding tissue, we cannot regenerate, restructure that in the lab. So it's possible that we're missing a very important information. And that's why, initially sensitive in the lab, then we go to the patient or to the animal model, and the results are completely different. And of course, there are external factors as far as measurements, errors, etc., done within the lab, which they cannot be easily controlled for. So these are the potential problems. So where do we go from here? So we like to know who benefits from treatment. We like to predict that. We like to avoid toxicity. And then if we can target certain proteins or certain molecules to improve the outcome, then that's fantastic. So there are a few things we could talk about. I briefly mentioned. So one of them is we talked about the microarrays, and it's only looking at the genes that are being expressed at the time. So whole genome sequencing of the cancer that might have a role. Very little is done about it, as far as I understand, especially in the setting of bladder cancer. So that might be one approach. Using xenografts, as Dr. Black would like to do, is and then doing gene expression profiling and try to manipulate the situation within an animal itself. So without altering the microenvironment is another option. And fewer models are available to use, the whole tumor, use a spheroid model to kind of try to keep in mind the whole microenvironment aspect of things. There are some other ideas available, which these guys have published 3 or 4 papers. These are from, I think they're from University of California, Davis. So the idea is good. The evidence is limited. So the idea is, okay, look, we use a carbon model in the cancer molecule with the C14, so we can follow what's happening to it. Give it to the patient in a small homeopathic dose, and then take a biopsy of the tumor subsequently. Then use this accelerated mass spectrometry, which can measure small concentration as low as 10 to the power of minus 20. So very, very small concentration. And based on that, for example, measure the DNA dimers and say, well, did we get enough DNA dimers? So based on that, would we be able to predict whether this cancer is going to go toward apoptosis and have good clinical response? And based on that, if we know the pathway involved with the drug delivery and metabolism, that's the hypothesis. We can see, for example, we have too much of this here, then probably this enzyme is lacking. So let's try to come up with a product that bypasses that. Or let's try to give the patient another molecule or another protein as a helper to try to overcome that problem. So the hypothesis makes sense. Because we're giving such a small dose, it's unlikely to cause chemoresistance, it's unlikely to cause toxicity, and by doing that, we can avoid overtreatment and unnecessary toxicity. So the idea is good, but very limited data available. So that might be something to keep in mind for future. So coming toward the end of the talk, so just a very brief overview. I couldn't talk about this and not mention these names. So this study probably was ahead of its time. This is quite way back, so 7-8 years ago. It goes back to clustering. We identify the targets. Now the next step would be, well, come and hurt it, come and block it in a way, either using interfering RNA or antisense oligonucleotide, etc., to try to switch off that transcription in an aim to overcome the chemoresistance. And Allen, Dr. Glebe, etc., they showed that in their paper. They had knocked that mouse, they gave him bladder cancer, they turned it off, and it responded very well to chemotherapy subsequently. There is phase 3 trials for lung cancer and I believe ovarian cancer running, so that might be an avenue. Knowing what I know, although so little it is, I think that might not be the answer though, because the cancer cells are clever. You block one pathway, they go through other pathways. So probably same way with chemotherapy that we target use multiple agents, probably the way is to target multiple genes. So this is a new paper, and these guys came and targeted a lot of anti-apoptotic genes plus survivin in an aim to improve the chemosensitivity, and that worked. But again, whether this is going to be the same result in the clinical series is remain to be seen. Now we briefly mentioned the extrinsic apoptotic pathway, and so when this intrinsic pathway is faulty, it might be possible to use an extrinsic pathway to directly push the cells toward death. Obviously, it has to be specific to the cancer cells, cannot cause death with other cells, but it might overall improve the chemosensitivity. So this is an agent that works on some of those extrinsic apoptotic pathways, like the trail ligand, trail receptor, etc. And it has shown promise, but again, limited data in urothelial cancer. So it's an exciting time. You know, we are understanding more. Hopefully we can identify the people who would benefit from certain regimen, and the guys who don't benefit, we might be able to target specific protein to overcome that problem. So future will tell which way we're going to go. Just like to, this mostly for residents. Look, when I went through the training, there was no mention of genetic of cancer in our books. I didn't know anything about it till I got here. Probably I wasn't a very good student, but regardless. The new chapter of the new edition of Campbell has a nice summary. It's about 30 pages. It covers the basics, so it gives you a good basic background. And you know, if you're attending talks or reading about for your exam or etc., it's a good platform to start with. I'll be happy to answer any question. Thank you.